Hello everyone, and welcome to our first lecture on immunity. If you look in your textbook, you will see that there are 2 chapters on the immune system, and the reason for that is because there are 2 main divisions of the immune system. And in this chapter, we will cover the first division, which is called the innate immune system. Talking about the immune system at this point continues our theme of talking about how we control microbes. In our first lecture, we talked about how to control microbes outside the body. In our second lecture, we talked about how we can control microbes inside the body with certain types of medications. And now we'll be focusing on talking about how we control microbes inside our body with our different types of immune responses. So like I mentioned on the previous slide, the immune response or the immune system can be broken up into 2 very broad categories. We have innate or non-specific immunity, and then we have acquired or specific immunity. This lecture will focus on innate immunity, and the next one will talk about acquired or specific immunity. On this slide, you just have a little bit in terms of the characteristics about both types of immunity, so you can kind of compare and contrast. You can do that now if you want to, so you know what to listen for in these next 2 lectures. And then you may want to do that again. You may want to come back to this slide after you've gone through both of the lectures so you can really look at these characteristics and make sure you're clear on what they are, what is unique to each type, what might overlap between the 2 of them, and how they all work together to make sure we have a functional immune system. So now that we know that there are 2 basic types of immunity, innate immunity and acquired immunity, we will go ahead and get into the details about the innate immune system. As always, you have your lesson objectives, so I recommend you look those over now, make sure you know what you're listening for, and then come back to them after you have listened to the lecture and make sure that you have completed these objectives. Innate immunity provides our body with a general way of defending itself against microbes. So most of the time, innate immunity works the exact same way every single time we are exposed to a given microbe or just microbes in general. And it is fairly consistent throughout humanity, meaning that all of us have the same basic innate immune system, or the same basic components of the innate immune system. And the way they work in my body, are the way they work in your body, are they the way they work in someone in the middle of a continent that we've never even been to. Basically, every single person on the planet, in the absence of some kind of genetic difference, has the same type of innate immunity. It's a general immunity because it responds to microbes the same way every single time that it is activated, which is pretty much all of the time because we are always in the presence of microbes. So no matter what microbes we are exposed to, whether they are ones we have seen before or ones that are totally new to us, our innate immune system responds the same way every single time. And the things that happen in my body with the innate immune system are the same ones that happen in yours, are the same ones that happen in your cousins, and everyone else in the entire world, basically. The main goals of innate immunity are to prevent entry. So that is the first goal of the innate immune system. And we prevent entry with different types of barriers. We have chemical barriers and physical barriers. If something makes it past those barriers, which things do, that happens every day to most of us in some way or another, whether we notice that or not, the next thing that happens is a series of steps that help immobilize the microbe, which means keep it in one location so it doesn't get deeper into our body, and destroy the microbe pretty much in the place that it entered, or at least hopefully near the place that it entered. The next thing that happens is once that microbe has been destroyed, there are certain cells that were involved in destroying that microbe that will then signal to the adaptive immune system to get ready to make antibodies. So the innate immune system, in addition to helping us deal with microbes here and now in this present moment, helps our adaptive immune system develop as well so that we can be ready to fight them off in a different way the next time we're exposed to that same microbe. The first line of defense in the innate immune system is composed of the barriers. The purpose of barriers are to keep microbes out of the body or out of specific cells. There are two broad types of barriers. The first one are the chemical barriers, and the second one are the physical barriers. Chemical barriers are found in various locations in the body. One great example are the barriers found on our skin. So our skin is slightly salty and slightly acidic, so it has a little bit of a low pH and a little bit of a high tonicity or osmolarity. And this keeps many different types of microbes from being able to successfully live on our skin. So it doesn't keep all microbes from getting on our skin. There are plenty of microbes that can live on our skin just fine and actually prefer that type of environment. But many microbes in the world are kept off of our bodies because of the nature of our skin. The fact that it is a little bit acidic and a little bit salty ends up creating a habitat that is kind of hostile for many types of microbes. We also find a chemical called lysozyme in many locations in our body. Many of our bodily fluids, especially things like tears and saliva, have this chemical contained within them. And the purpose of lysozyme is to break down and destroy peptidoglycan. That way, when bacteria get in our mouth or in our eyes, we have a type of defense mechanism. We have this barrier that will help prevent them from getting any farther because the lysozyme will start to break down the cell wall of bacteria. Other types of chemical barriers are the defensins. Defensins are a group of proteins that are found primarily on the surface of our skin. And what they do is create holes in cell membranes. So if we have a bacteria or a helminth or an algae or a fungus or a protozoa, any type of microbe really, even a virus if it has an envelope technically, and it ends up on our skin, these defensin proteins will attach to that lipid bilayer that microbe has and start to create holes in it. And especially for everything except viruses, that cell membrane is really what is keeping them alive. So if you destroy the cell membrane, everything that is outside the cell comes in, and everything that is inside the cell goes out, and you start to destroy the organism that has this hole in their cell membrane. The other main type of barrier that we have is the physical barrier. Our biggest physical barrier is our skin. So while there are microbes that live on our skin and just slightly below the surface of our skin, most of those microbes are mutualists with us, meaning they help us out, we help them out. Many of them may be commensalistic with us, meaning that they get a benefit, but we're not really harmed by them. So even though the skin doesn't completely prevent all microbes from living on the surface of our bodies and just slightly below that surface, what it does is prevent most of the pathogenic microbes that we encounter on a day-to-day basis from getting inside. So we do have microbes that live on our skin, but those microbes, in addition with our skin, actually do help create this barrier that prevents the other microbes that we encounter on a day-to-day basis from getting inside. The shape of our cells and lacking certain types of receptors can also be barriers. Many microbes need to actually be inside, not just our bodies, but inside specific cells to be able to do damage. One great example of this is the protozoan that causes malaria. So the name of that protozoan is Plasmodium. And I'm actually just going to give the genus name because there are a couple of different species of this protozoan, and we're just going to talk about it in general. But Plasmodium is a protozoan that needs to get not just inside our bodies, but actually inside our red blood cells to be able to cause disease. So if this is a standard red blood cell here, plasmodium will get inside our body, and it won't just get inside the body. It has to actually get inside the cell in here to start to cause problems. So this is our plasmodium, a parasite that is sitting inside the red blood cell, and although it's pretty happy, it's causing some problems for us. However, if we have a variation in our cell type called sickle cell trait, then our blood cells are not shaped in what we would consider kind of the wild type red blood cell. So the wild type red blood cell is what we call this biconcave disc. And when someone has something called sickle cell trait or sickle cell anemia, at least some of their red blood cells are shaped a little bit differently. They're kind of elongated and stretched out. And although this can cause some health problems for the people who are carrying this type of red blood cell, one of the benefits of it is that this protozoa that causes malaria cannot fit inside. So this guy will get inside this person's body, and it is not going to be able to get inside their red blood cells, and this person is not going to get malaria. So the shape of certain cells can be barriers for certain types of infections. This doesn't work for every single type of infection, but for many types of microbes, not only do they have to be inside our body, they have to get inside specific cells. And if someone has a cell shape that is not conducive to that bacterium or protozoa or fungus being able, or virus even, being able to get inside the cell, then that person will not be able to become infected with that particular microbe. The same basic idea kind of applies to the lack of specific receptors. This is especially true for viral infections. Remember, viruses have to be able to attach to a host cell using a receptor that's found on the surface of that host cell. And if that host cell is lacking an appropriate receptor, then the virus cannot attach. So in this case, this virus can fit very nicely into this receptor and will then be able to get inside and infect that person's cell. But let's say that receptor was shaped just a little bit differently or just didn't exist at all. So if it was shaped differently, then the virus would not be able to attach to it. Or if that receptor was completely gone, so just crossing it out, if that receptor is completely gone, the virus has nowhere to attach to, and if it can't go through the adsorption stage, it can't go through any of the other stages of replication and can't make the person sick. So our skin obviously is the biggest physical barrier we have, but the shape of our cells inside our body and the specific receptors that we have or don't have on our individual cells can also be barriers to infection. Now, if something does manage to get through our barriers, and that actually happens on a fairly regular basis, so most days of our lives, things will get through our barriers at least a little bit, then our second line of defense becomes activated. Our second line of defense has multiple components to it, and the first component we will talk about is inflammation. The purpose of inflammation really is kind of twofold. It is to trap microbes and slow them down, and also to mobilize and attract other immune cells from our bodies. So trapping microbes helps keep them localized, preventing them from spreading through the rest of the body and causing more damage as they go. And then attracting these other immune cells helps those microbes become destroyed, hopefully just in that local area, again, so that they don't spread and cause further damage. In terms of the process, we've got kind of an overview of this process right here. It essentially starts when microbes enter bodily fluids. So here this person has got a cut on their skin and some microbes have come in to their body. When those microbes come into the body and when that injury occurs, a whole host of different types of chemicals, most of the time proteins, are released, and they cause a cascade of signaling to occur in the body. So these signaling proteins are usually referred to as cytokines, And I'll talk about cytokines more later in this lecture as well as the next one. But basically, cytokines are just generalized signaling proteins that tell our body something is wrong and you need to behave in a different way to take care of this wrong thing that has happened. So once these cytokines are released as a result of the bacteria coming in or other microbes coming in and the damage to the skin, then our body responds by increasing the amount of fluid that is in a particular area. So more blood cells get sent over to that area, more fluids get sent over to that area, and that area starts to swell. As those fluids are being brought over and those extra cells are being brought over, what they do is start to release some proteins that causes kind of a mesh or net-like structure to form. Certain red blood cells called platelets will come over and start to join together and help close off the wound. And other types of cells, many of them white blood cells, will come over to the area as well and start to engulf this microbe that has invaded our bodies. So they engulf the microbe, they take it into themselves, their bodies, they start to destroy it, and then when that microbe is gone and those cytokines are no longer present, then the fluid can be removed, the cells can be removed from that area, the wound can continue to heal, and everything can go back to normal. At the same time that inflammation is occurring, another one of our second line of defense mechanisms is activated as well, and this is phagocytosis. Now, phagocytosis can be activated independently from inflammation, but oftentimes the two go hand in hand. They don't have to, but most of the time, if you find inflammation, you're going to find phagocytosis, and often if you find phagocytosis, you will find at least some markers of inflammation as well. But the purpose of phagocytosis is to engulf and destroy the invading microbes. So that's the whole goal of phagocytosis. It is just for our body cells to grab on to these invading microbes and start to destroy them. Technically, it happens in 3 steps. The first step is what we call chemotaxis, binding, and ingestion. In this step, our body cell, and there are several different cells that actually can go through phagocytosis. Right now, we're just going to call these just kind of our general phagocytes. There are multiple cells that can do that. We'll get more into that in the next lecture. So this phagocyte, the cell that is capable of engulfing these microbes, is going to move over to the location of the microbes. They are going to bind to the pathogen and start to engulf or bring that pathogen inside themselves. The next stage is called the phagolysosome formation. Within this phagocyte, there are organelles called lysosomes. Lysosomes contain lysozyme, and lysozyme, as you remember from a previous slide, is geared towards destroying peptidoglycan. So the lysozymes that are inside the phagocyte are going to move over to this structure that is called the phagosome. The phagosome is kind of a temporary vesicle that contains whatever it was that was brought into the cell. So the phagosome is this temporary vesicle that contains the microbes or the microbe that was engulfed, and the lysosome is a vesicle that contains lysozyme. So these two vesicles are going to join together. Basically, the lysosome is going to attach to the phagosome and kind of distribute all of that lysozyme into this vesicle that is now merged together. So now we have a situation where the phagosome and the lysosome have joined together, and that is now called the phagolysosome. And the lysozyme has been exposed to the bacteria, or the bacteria have been exposed to the lysozyme, and it can start to break them down and destroy them. Once that begins to occur, then the next phase of phagocytosis begins, and that is destruction and elimination. So in this phase, we have the phagolysosome that is going to be working on destroying the bacteria. The bacteria are starting to get destroyed, and all of their individual components are kind of amassed together in this one big ball of protein and lipid and DNA and broken up bacteria. And the lysozyme can then be recycled. The cell will get rid of that, put that back into a lysosome, and the waste product of the bacteria can be gotten rid of. So that would be the elimination stage. So during the destruction stage, the bacteria are all torn apart. They're kind of disassembled by this lysozyme. And then those lysozyme molecules can go on to be reused, while that broken up bacteria, that big tangled mess of everything from proteins to lipids to nucleic acids, etc., can be eliminated by the phagocyte, and the phagocyte can go on to repeat this job somewhere else. Along with inflammation and phagocytosis often comes a fever. Not always, but oftentimes. The purpose of a fever is to increase the temperature of the body, either the entire body or a specific location in the body, so that metabolism will speed up and microbial growth will be slowed. So sometimes when we have a rather large infection that is spreading throughout our body, our entire body will increase in temperature, which will help to speed up our own metabolic reactions and oftentimes help slow down the growth of the microbe. Or sometimes if we have more of a localized infection, we just find that heat in that specific area. So for example, let's say you step on a nail or a thumbtack or something sharp and you get an infection in your foot. If your body's handling it well, you may not run a fever throughout your body, but you may notice that there's some heat in that specific area, right? It gets a little bit red, it gets inflamed, there's extra fluids there, it might get a little bit hard because of those extra fluids in that area. And that heat there is really the body's defense in terms of trying to increase the metabolism to make sure that things like phagocytosis can go faster, and also hopefully help slow down the growth of this microbe. Because as you remember from last unit when we talked about things like cardinal temperatures, sometimes even just a slight increase in temperature can be enough to slow the growth or potentially even kill a microbe that is very temperature sensitive. Another aspect of the second line of defense for the innate immune system is something called the complement cascade. Now, the complement cascade is actually very complex, and there are multiple pathways. So there's the classical pathway, there's the lectin pathway, and then there's the alternative pathway. We're just really going to focus on the classical pathway for the most part. So I'm showing you these so you can see that they exist, but don't worry about them too much for now. What we just want to focus on primarily is this classical pathway. So the purpose of the complement cascade is to create a series of proteins that can be used to stop the growth of microbes. It works in multiple different ways, depending on exactly which pathway is used and depending on other factors that are occurring in the body. But the one we're going to focus on is the membrane attack complex. So there are some other things that go on with the complement cascade. We're not going to focus on those quite as much. What I want you guys to pay attention to are the classical pathways and something we call the membrane attack complex. So you know these other things exist. You're free to read more about those or ask me questions about those if you want to. But really what you're going to need to know for exams and stuff are the information about the classical pathway and the membrane attack complex. So the complement cascade, at least in this sense for the classical pathway and the MAC or membrane attack complex, the purpose of that is to help create holes in microbial membranes. The first step is what is called initiation. During initiation, the body recognizes that there is a foreign invader present, and it begins to produce a series of proteins. It starts by producing this protein called C3. From there, additional proteins are produced. So C3 breaks up into what we call C3A and C3B, and this causes another series of chemical reactions to occur. As all of these proteins are being produced, once there are enough of them that are ready to be used, the next step called polymerization occurs. Polymerization is when all of these proteins that have been produced in the previous steps are going to join together and form what we call the membrane attack complex. Once polymerization has occurred and the membrane attack complex has been created, and remember what this membrane attack complex is basically just a bunch of proteins that have joined together, then the next stage, or the final stage, which is called membrane attack, can actually occur. What happens here is that this MAC, this membrane attack complex, inserts itself into the cell membranes of microbes. Whether that microbe is a worm, whether it is a protozoa, whether it is a bacteria, whether it is a fungus. It may even work on enveloped viruses. But basically what ends up happening is it kind of creates a situation as if you were sticking a pin into a balloon. The membrane attack complex inserts itself into the membrane, and it causes this big hole to form inside that microbe. Then, as we've kind of talked about this before in multiple different situations, everything that is inside the microbe goes out, anything that is outside of the microbe goes in, and the microbe gets destroyed. The last aspect of the second line of our innate immune response that we will talk about are the interferons. Sometimes interferons are abbreviated IFN, sometimes INF. I wish I could tell you for certain which one to look out for, but it kind of seems to depend on what resource you're looking at. I tend to say IFN, so most of the time I abbreviate it as IFN, but I have also seen it as INF, just so you know. But interferons basically are proteins that have the purpose of signaling to body cells that there is a virus present and they should not let that virus replicate. So the overall purpose or the overall goal of the interferons are to prevent the virus from spreading throughout the body. They can also be used to help inhibit the growth of other microbes or other types of cells as well, but we are going to focus on them in the context of viruses. The way interferons work is that as soon as a virus attaches to a host cell, the host cell recognizes that that virus is there, and the host cell wants to warn other nearby cells that there is a virus around. So what this cell does is create interferon, which is this protein that will then be released and will be sent over to neighboring cells. Interferon attaches to these neighboring cells, and this causes a couple of different things to happen. So one of the things that interferon can do is tell neighboring cells to block off their receptors if possible. Basically, these proteins will say to this neighboring cell, hey, there's a virus present, and if you could remove your receptors or just block off your receptors, that would be great. That way, no more viruses can attach to you, and you won't replicate them and spread the virus. Another thing that interferons can do is tell neighboring cells that might have got infected by the virus and they didn't really know it, that, hey, there is a virus around. You might want to check and see if you're infected with it. And if you are, start to go through the process of self-destruction. That way, you don't end up spreading the virus throughout the rest of the body. And then finally, another thing that interferons can do is help recruit other types of defense cells to the particular area so that these defense cells, things like phagocytes and other types of white blood cells, can help get rid of the virus. So now we've gone over the highlights of the innate immune system, and one of the things I wanted to do for you was create this summary slide because there is a lot of information about the immune system, and sometimes it can get confusing. So just to give you a summary of what we talked about, the innate immune response is present from birth. Basically, we're born with it, and it's active our entire lives. Most days, we have at least a little bit of the innate immune response going on because most days we are exposed to microbes. The innate immune response is nonspecific, meaning it basically just behaves the same way every single day of our lives, every single time we're exposed to microbes. It is the same for all humans, and I put that little star there because it's the same for all of us unless someone has a genetic difference in part of their immune system. And it works the same way all of the time. So from the moment you're born until the moment you die, things like inflammation, phagocytosis, complement cascade, interferons, they're going to work almost the exact same way your entire life, every single time they're activated. The innate immune system makes up our first two lines of defense. So the first thing it tries to do is keep things out, and the second thing it tries to do is trap and destroy microbes locally so they don't spread through the body. Most of the time, the aspects of the second lines of defense work together. So typically, inflammation and phagocytosis go hand in hand. Is it possible to have inflammation without phagocytosis? Probably. Is it possible to have phagocytosis without inflammation? Yes. Is it possible to have those things without a fever? Yes, it is. But oftentimes, all of these things kind of coordinate their efforts to create a multifaceted or multi-pronged approach to getting rid of a microbe that has invaded us. Now, the last point on the slide is kind of a little bit of a new concept, and the reason I'm introducing it here is to help you get ready for the next lecture. But some of what is happening during things like inflammation and phagocytosis and fevers, etc., and the complement cascade is helping prep the adaptive immune response to develop. So in our next lecture, we're going to cover the adaptive immune response and talk about how that one gets going. And one of the things to remember is that the innate immune response typically helps the adaptive response get going.